in your head, whatever, as a similarity or difference, some key concept that answers this question. Janusz.
They're both passive transfers.
Okay. So a similarity, passive. They're both passive. CJ?
So for an ion channel, like many ions can pass through when it's in the open conformation, but for facilitated with like transport molecules, only one ion can move per conformational chain.
Right. So let's so a difference. Ion channels. Many ions per conformational change. Uniport. One solute could be an ion. Maybe it's two sometimes per conformational change. Just out of curiosity, is the Greek delta, for me, I just automatically see that as change. Is that true for most of you? Okay. That's what that triangle is. It's a Greek delta, delta G, right? You've seen it before. Okay, yes?
I was going to say they both go through conformational changes to help amphipathic.
Okay, yeah. So a similarity is they undergo conformational changes. Excellent. Sophie?
They have binding of solute,
Is that a similarity or a difference?
similarity.
Yeah, so they both have binding sites.
They both go down concentration.
That's pretty much a definition of passive, but good. The solute moves down a concentration gradient. How many of you know the upside down triangle is the symbol for a gradient? No? Okay, so I'll do that. Okay, any others? So, Abby?
Binding and unbinding interactions are faster for ion channels.
Good. So they have binding sites, but the binding and unbinding reaction rates are faster for ion channels. Any others? I can think of one really obvious, you'll go like this, similarity. Yeah, Sarah.
I was going to say solute type.
Hmm?
Solute type, I don't know like how well you can say it, but like a channel most commonly uses ions, but a carrier more commonly has like larger uncharged
I
molecules.
buy it. I think that's true. So the difference is that channels are almost always ion channels, with one key exception. There is a water channel. So I haven't told you about aquaporin, but it's a water channel. But most of the channels we'll talk about are ion channels, and many of the uniporters are ions or small molecules. So that's true. Robbie?
Both selected for a single molecule.
I'm sorry, say again?
They're both selected for a single molecule.
Very good. So they both have selectivity for their preferred transported ion or small molecule. The binding sites are selective for the selected molecule. So the glucose transports glucose, but sometimes transports some other sugars, but it doesn't transport amino acids. It's selective for glucose and a few other sugars. The sodium channel is selective for sodium over potassium and calcium. That's selectivity. Good. And
Protein membrane protein.
they are all membrane proteins. That was what I was thinking of. The obvious similarity. Good. Any others? The next slide has my answers. Let's just see how well we did together. And this list might actually be a better list than the one I came up with last night when I was just coming up with answers, because I was doing it fast. Let's just see. Okay, so we got the idea of passive, high to low concentration, ion carriers, 1 to 2 ions per conformational change, we got that. Ion channels are passive, thousands, hundreds or thousands per conformational change, we got that. Similarities, both passive, binding sites, conformational changes, selectivity. You got the, Robbie, that was your, Robbie remembered that they are selective, and I forgot about it last night, which is weird because I've studied selectivity of ion channels my whole life, my whole scientific life. Differences. 1 or few ions per conformational change versus hundreds or thousands. We did a better job with differences. What do we?
And the rates, we had the rate as well.
We had what?
The rate.
Oh yeah, rates of binding, yeah. So, congratulations, you came up with an excellent selection of similarities and differences. Okay, now I'm going to spend about 10, 15 minutes talking about active transport. So we're transitioning to active transport, pumps. And then we're going to transition to our about half an hour of working on the problems. So that's coming up. So pumps harvest energy from some source and pump an ion or a solute up the concentration gradient. So this is not downhill movement, this is creating the gradient. Well, you can't create a gradient is a form of energy, and you can't create energy unless, well, second law of thermodynamics, we only convert energy. So we convert energy from some source to create these ion gradients. That's what an active transporter does. A primary active transporter takes the energy from ATP and converts it into an ion gradient. So the sodium-potassium, ATPase, you're all familiar with ACE, tells you it's an enzyme. So this enzyme hydrolyzes ATP, and in the process of hydrolyzing ATP, it pumps sodium out and potassium in to the cell. This is true, every single cell in your body does this. So what this does is it means that the sodium concentration is high on the outside and low on the inside because this ATPase pumped sodium out. And the potassium concentration is high on the inside and low on the outside because the ATPase pumped the potassium in. And there are leaks and there are channels that are dissipating this gradient, but your cells have a lot of this, and if your cells are healthy, they have a lot of ATP. So your cells are constantly maintaining this gradient. I don't want you to memorize these numbers. I don't want you to memorize these numbers. I want you to understand the concept of the sodium-potassium ATPase converting the energy in ATP into a solute gradient. So,
Why wouldn't you have the free potassium ions, I guess, phosphate ions? So when the ATP just says
Where does the phosphate go? It's in the cell, and it will be used by the mitochondria to remake, so ADP and phosphate are the substrates for the ATP synthase in the mitochondria to remake ATP. So the phosphate and the ADP will be recycled, and the mitochondria that you learned about in cell phys will turn that back into ATP as long as there is a fuel source. So here is a blobogram of how the sodium-potassium ATPase works. And I'll walk through this because it illustrates a number of the concepts that I want you to know, to be thinking about whenever we talk about movement of solutes via active transport. So here, in this case, the intracellular solution is up and the extracellular solution is down. You always have to get oriented. Here's the membrane. Here's the protein. We can start anywhere in this cycle because it's a cycle, but let's just start at the beginning. So sodium ions bond. So diffusion and binding. ATP has bound. Okay, so now, let's tell the story as a group. Instead of just walking through this. What happens between here and here? Mike?
Looks like those 3 sodium bind, and then there's a conformational change that opens up the channel.
Okay. The binding of the sodium caused a conformational change of the protein. It's not a channel. It looks like a channel. This is a pump. So let's just make sure we use the right language. If you tell me the sodium-potassium channel, I'm going to say, wait a second. If you understood that this is an active transporter, you wouldn't use the word channel. So we have to be precise about our use of language. If you say sodium-potassium channel, when you're talking about the sodium-potassium pump or the sodium-potassium ATPase, I will get confused, I will think you're confused, and we're not communicating well, and I'll take a point off of a quiz or an exam. Okay, so you're right, a conformational change of the pump. What else happened? No.
ATP is hydrolyzed and ADP dissociates where the PI is bound to the
So ATP has been hydrolyzed, and this phosphate is now covalently attached to the protein. What do we call that process? When ATP is hydrolyzed, Olivia? No? When ATP is hydrolyzed and the phosphate is now attached to the thing that the ATP used to be just sort of loosely associated with. Olivia?
Phosphorylation?
Phosphorylation. In this process, the pump is phosphorylated. So the binding of the ions and the phosphorylation of the protein causes this conformational change. Actually, it's the binding of the ions that cause this conformational change, and it's the phosphorylation that causes this conformational change. Okay, what is this illustrating?
The release of those.
Unbinding and diffusion of the sodium ions. They notice that they bound to the inward facing conformation, and they unbound from the outward facing conformation. What do you think this conformational change, let's go from here to here, what did this conformational change do to the affinity of the sodium binding sites of this protein? I need to hear louder. Decreased it. High affinity binding in the inward facing conformation. Low, that's what these low affinity binding in the outward facing conformation, and that's why the sodium is released. The conformational change changed the side to which the sodium is, the sites are exposed, and the affinity of those binding sites. So now, if we had a lot of sodium here, would sodium bind very actively to these sites? No, because they have a low affinity. We'd have to have a boatload of sodium on this extracellular fluid, and that's non-physiologic. Okay, so now we have an outward-facing site with low affinity sodium binding sites, but high affinity potassium binding sites. So, okay, this is, I just gave it away. What happens here? Potassium binds to the high affinity binding site. Potassium from the outward facing conformation and the high affinity binding sites allow potassium to bind, even though the potassium concentration outside is not very high. What do we call this process? Dephosphorylation. Right, so phosphorylation and dephosphorylation. Binding and dephosphorylation trigger another conformational change back to the original conformation. High affinity sodium binding sites, low affinity potassium binding sites. Potassium is released because the affinity is low to the inside. ATP binds because if the cell is healthy, there's lots of ATP. And we're back to where we started. So you should be able to tell the story of the sodium potassium ATPase. That's a homework assignment or something. Just be able to tell the story. Sodium binding, ATP binding, conformational change, phosphorylation, more conformational change, sodium release to the outside, potassium binding from the inside, from the outside, dephosphorylation, conformational change. That's the story of the sodium potassium pump, sodium potassium pump. There are proton pumps. We're not going to talk too much about those, but they exist. You've heard a little bit about them in self-phys. You've heard about the proton ATP synthase, the proton-driven ATP synthase. Well, that's a proton pump running in the direction of using the proton gradient to make ATP. But sometimes we use ATP to make a proton gradient. It can run both ways. All I want to do is remind you that there are proton pumps. So we're only going to have three of them. We've got the sodium potassium pump, proton pumps, and before I leave the slide, what do these blobs, what is this colorful blobogram conceptually trying to illustrate? It's telling us that this is this is running an ATP pump, ATP-driven pump, because if it were an ATP synthase, this arrow would be going the other way. But what's the concept of the blobs? I'm thinking protein structure. So here, we don't see multiple blobs. And here we do. What is that concept? Subunits. Almost all of these proteins are formed by multiple subunits. So even though we don't see them here, they exist. Something like this. And there are calcium ATPases. So we have the sodium, potassium, proton, and calcium ATPases. And those are the three most important primary active transporters. The calcium pump. So, and we're going to talk about, well, let me go to the next slide. No, I'm going to come back to this. There are going to be two types of calcium ATPases, two pumps. One is in the sarcoplasmic reticulum membrane, and we call that one SERCA, or sarcoplasmic endoplasmic reticulum ATPase. Calcium, I forgot, the most important. Calcium ATPase. This is a calcium pump in the sarcoplasmic or endoplasmic reticulum. We'll come back to the definition of that membrane. It's an intracellular membrane compartment. You may remember it from cell phase. The other calcium ATPase is called PMCA, for plasma membrane calcium ATPase. It's in the surface membrane of the cell, like the sodium potassium ATPase. Both of these pump the calcium out of the cytoplasm. So we'll see that in an upcoming slide. I want to introduce, so this is taking longer than I thought, but that's okay. I want to introduce you to the concept of a different active transport. So we've talked about primary active transport. Now we're going to talk about secondary active transport. It's active, but it's not primary, it's secondary. What's the difference? In secondary active transport, a solute gradient is the source of energy to create another solute gradient. So, for example, here, sodium, the sodium gradient, was created by the sodium potassium ATPase. Primary ATP was the source of energy. Sodium is in high concentration on the outside and low concentration on the inside. That's a form of energy. The gradient is a form of energy. That was created by the conversion of ATP. So that source of energy can be utilized by secondary active transporters to create a different gradient. So here, the sodium gradient is coupled to the transport of another solute, and when sodium is going down its concentration gradient, because that gradient was created by the primary active transporter, sodium is going down its conformational change. That provides energy that moves a different solute up its concentration gradient. And so you'll see when we get to the kidney, it's going to be very important. We're going to see a sodium glucose co-transporter. The sodium gradient and the glucose are obligatorily coupled. And so when sodium is moving down its concentration gradient, glucose is moving up its concentration gradient. The transporter is pumping glucose using the energy from sodium. So we have two types of secondary active transport. We have co-transport, where the two solutes, the sodium and the glucose, say, are moving in the same direction. And we have exchange, or antiport, where the two solutes are moving in the opposite direction. So this is the sodium-calcium exchanger, which we're going to talk about in the heart really soon. So let's start here, because I just find it easier to start here. Here's the outward-facing configuration. This is a blottogram. It's a little bit more angular than typical, but sodium binds. Conformational change. Sodium unbinds, because the affinities are low. Calcium binds. Conformational change triggered by calcium binding. Outward facing. Calcium unbinds and sodium binds. So sodium is flowing down its concentration gradient from outside to inside. And that energy is harvested by this protein, because when sodium goes down its concentration gradient, it creates a situation that calcium can bind from the inside, high affinity calcium binding, conformational change, because calcium bound, now it's facing outside, conformational change changed the affinity of that binding site, so calcium is released to the outside. So this is a sodium-calcium exchange. The next slide shows calcium homeostasis. We're going to need this for the heart. The mitochondria are important, but I'm going to skip them to simplify the situation. So this is the slide you have on your handout, but it's the same slide as this. It's just, we're going to skip the mitochondria. So I'm going to introduce the players. You're going to use this in your problem set. So let me just introduce the players and not screw up the problem set. Plasma membrane calcium ATPase. Primary active transport pumps calcium out of the cytoplasm, out of the cell. Circa, primary active transport, pumps calcium from the cytoplasm into the sarcoplasmic or endoplasmic reticulum. Sodium-calcium exchanger uses the sodium gradient, which isn't set up on this slide, but you know it's there, to pump calcium out of the cell. So before we go into working on problems, where in this cartoon is calcium concentration low? Cytoplasm. Does anyone remember from maybe just from reading or have a sense how low is low in this case? About 0.1 micromolar in a resting cell. So 0.1 micromolar. Where is calcium high? Outside? And where else? In the SR or ER, in the endoplasmic reticulum. And the calcium concentration there is millimolar. So millimolar, micromolar, there's 1,000-fold. Couple of millimolar, 0.1 micromolar, so it's a 10,000-fold gradient in calcium concentration. That's a very high gradient. We'll talk about these other proteins, the calcium channels in the endoplasmic reticulum later. Let's not get confused by those. But we have channels. We have calcium channels in the surface membrane, and we have calcium channels in the endoplasmic reticulum membrane. And so these channels are as far as calcium is concerned, are a leak, right? Calcium is flowing in, down its concentration gradient, but the pumps and the transporters are maintaining a low resting calcium concentration of sub-micromole. Let's skip the clicker. Let's get to work. So let's take a break now. Take a 5-minute break and come back in here and let's plan to work on the problem set for about a half an hour. You can work together, but I want you to maintain safe practices with regard to distancing. So you feel free to talk to each other, but don't huddle over one piece of paper.